start with the central dogma of molecular biology. Now, what is the central dogma of molecular biology? Now, we know in molecular biology or the synthesis of protein that starts with the DNA. The DNA replicates, and this DNA contains the information which is required for the protein synthesis, which leads to the synthesis of various enzymes. Now, this information is given to the transfer RNA, sorry, the messenger RNA by the DNA in a process called as the transcription. Now, this information is carried from the DNA to the cytoplasm by the messenger RNA. So that is what is called as the transcription. Later on, that information which has been brought out by the DNA in the cytoplasm by the messenger RNA is converted into the language of protein by the transfer RNA in a process called as the translation. Now, normally we know translation is we are changing that language from one language to another language. So whatever the language of DNA and RNA is there, that is now changed into the language of protein. So transfer RNA brings that amino acid sequentially as coded in the DNA, and that code of DNA is brought out by the RNA. So first is your transcription followed by translation, and what is involved is the DNA, RNA, and finally what is formed is the protein. This is what is known as the central dogma of the molecular biology. So it involves DNA, RNA, the proteins, and the two processes of transcription and the translation. Then we have the Chargaffs rule. Now, Chargaffs rules states that now we know that in DNA you have two different types of proteins, the purines and the pyrimidines. Now, according to these Chargaffs rules, any cell of any organism, it has these base pairs, the pyrimidine and the purine, in the ratio of 1:1. This ratio never changes. So if you have a certain amount of guanine, you will have the same amount of cytosine. If you have a certain amount of adenine, then you will have the same amount of thymine. Now, this pattern is found in both the strands of DNA. So they will be in equal proportion or equal number or equal quantity. That is the purines and the pyrimidines. And this was discovered by an Austrian chemist, Erwin Chargaff, in 1940. And so it is known as the Chargaff's rule. The next is the C-value paradox. Now, what is a paradox? Now, paradox is a condition which makes us something difficult to understand, or which is difficult to understand or predict or interrelate. So, here, how can we count for the amount of DNA in terms of known function? So, the amount of DNA which is present in our genome is much more than that of the known functions present in our body. So, if you have similar organisms, they show a large difference in their C values. So, amount of genome in DNA in complex eukaryotes is larger than the amount needed to encode proteins. So, you have 30,000 to 50,000 genes in a mammals, but their genome size or the C value is 3 into 10 raised to 9 base pairs. Now, this can be explained by the fact that most of the part of the DNA is not coding for anything. So you can explain it likewise. So if suppose I put some certain things in my pocket. So I have certain paper chits, blank paper chits, certain old bills, and along with it, I also add certain currency notes. Now, any person certainly would be interested in that currency notes. So you compare the currency notes with the genes which are coding for certain things in that particular organism. And now the total content, whatever if it is present in my pocket, you compare it with their genome size or the C value. So my pocket is full with various papers along with the currency notes. So that becomes your C value. But when any of you remove that from my pocket, what will be useful for you is that currency notes. Other material will be useless, totally useless for you. So that paper, that sheet, the blank papers, everything will be of no use. Similarly, in the C value also, there are certain base pairs or certain genes which are coding for certain things in that organism. And remaining material is useless. So that is what is called as the C value paradox. So this we have already seen. The only part which remains of the DNA is the Watson and Crick's model of DNA. So you have the DNA which is made of two strands, and these two strands, they are twisted around to form a right-handed helix. The two strands are anti-parallel. Anti-parallel, the parallel, consider two snakes with their head lying in the same direction and tails in the opposite direction. You have the heads of this snail side by side and the tail of the snakes side by side. So they are said to be parallel. Now you turn one of the snake in the opposite direction. So you will have the head of the snake, and side by side you will have the tail of the another snake. Similarly, to the opposite end also, you will have the tail of one snake, whereas on the side you will have the head of the another snake. And that is what is called as the anti-parallel. So you have 3' end of one strand, whereas 5' end of the another strand. So here you have the 5' end, and here you have the 3' end. Here you have the 5' end, opposite to that you have the 3' end. And this is said to be anti-parallel. So two strands, they are anti-parallel. 3' end, it has a free hydroxyl group. This is the hydroxyl group, whereas the 5' end, it has the free phosphate group. Now the sugar and phosphate is polymerized, forming a nucleotide, and it forms a backbone. Whereas nitrogenous bases, they are stacked inside, one above the another. You have approximately 10 bases per turn. The asymmetrical spacing of the sugar phosphate backbone generates major groove and the minor groove. You have one major groove and the one minor groove. Now, major groove where the backbone is far apart, and in the minor groove, the backbone is very close together.